So I think.
Okay, so please open the starting slide. I will shortly introduce you, and then you will start. Okay, so we begin our seminar. It's a great pleasure for me to introduce Jean Clairambault from Paris, from Inria, senior researcher Inria, and also from University Paris. Well, before it was Paris 6, now it's Sorbonne University from Paris. And the title of Jean's presentation is Plasticity in Cancer Cells and Emergence of Drug-induced Drug Resistant, What Consequences for Therapeutics. So Jean, please, we are attentively listening.
Спасибо, Vitaly.
Yeah, by the way, sorry, one more question I forgot. So would you mind if we ask questions during the talk, or better to keep them when you finish?
You can ask during the talk, and if they are in Russian, I hope you will be able to translate, because I'm not sure to be able to understand the questions in Russian.
Yeah, for our listeners, I would like to say that Jean speaks very well Russian.
Not very well.
Not very well, okay. Not very well Russian.
Только немножко, к сожалению.
Немножко, к сожалению. And okay, we can use also this occasion maybe to ask something in Russian and to discuss a little bit. So, Jean, please, you can begin your presentation.
Thank you, Vitaly. So this presentation, this talk I will give now to you is about a question that came little by little when, with Benoît Pertam and others, we began to consider the question of resistance in cancer treatments. And little by little, it came to me that, in fact, such drug resistance is a kind of plasticity in cell. And so little by little, I came to things that I will present to you now that are not only mathematics, but related to evolutionary biology. But be sure that after some time, I will dive into mathematics. So the cancer is a disease of multicellular organisms. This is not always stressed enough by many people who are still working in single cells trying to find determinants of cancer. And in fact, you can find some, but you will never find all of the determinants of cancer if you consider only one single cell. It's not only a question of cell populations, but also of multicellular organisms. Cells in multicellular organisms are completely differently organized from the situation of, for instance, bacterial cell populations, for their growth, for their ability to change in the changing environment, etc. So for me, and I will try to develop this later, cancer is a localized loss, anatomically localized loss of cohesions between cells and tissue within a multicellular organism. Loss of control and differentiations is even, to my mind, maybe it's simultaneous to other people, prior to uncontrolled proliferation. And this is related, in my interpretation, to the atavistic hypothesis of cancer, which was first put forth in recent publications by Paul Davies, Charles Lineweaver, and Mark Vincent. And it asserts that it is a reverse re-evolutionary origin that gives rise to the emergence of cancer populations. That is backward evolution, reversing towards not only, not completely to single cell organizations, but to incomplete multicellular organizations. And it has been documented not only at the gene level by people in this Australian team, Triggers, Good, and others, but also earlier by people who showed by phylostratigraphic analysis that the genes that are disrupted in cancer are actually those genes that are linked to the evolution of multicellularity, starting from unicellular genes. So the natural question, if you consider valid all these assertions, is what coherence or cohesion? I will use coherence for signals and cohesion for tissues, but this can be discussed. Within and between cells and tissues made of inner multicellular organisms, why and how is it disrupted in cancer? This is a very general question, which relates, in fact, which is related to the kind of philosophy of cancer that I may speak about, but I will essentially speak about mathematics after some introduction. So when I speak about evolution, I can speak about evolution of cell populations in an organ or in a petri dish. This is a lifelong evolution, but it's different from the great evolution that has designed multicellular organisms in the course of billions of years. And when I referred lately to fast reverse evolution, it's on smaller time windows, but it relates to this change of time scale that inscribes itself in the time scale of the great evolution of billions of years. But it happens by a deregulation of the control of differentiation and proliferation at the scale of a human disease. And this may explain transient or established drug resistance. And intratumor heterogeneity, that is between cell phenotypic variability, is a consequence of such deregulation of differentiations, and it's a relevant setting to represent evolution towards drug resistance in tumors. If there are questions, I can hear that there are some bells that ring, but
no, in fact, sorry, John. No, in fact, it's not questions, but it's people coming to the audience. This is what. If there are questions, and so people can raise hands, okay. I will see it on my screen, and then I will ask you.
Okay. Sorry to have interrupted.
No, sure. Please continue.
So plasticity in cancer cells, which I define by a propension of epigenetic nature to reversal to a de-differentiated status that is going in a reverse direction from the point of view of an evolutionary tree, has as a result adaptability, in particular, of cancer cell populations to reversibly resist abrupt drug insult as a sharp stress response. So you may have heard about cellular stress, which includes a variety of cell responses to deadly insult or possibly deadly insult coming from the environment. And for me, such cellular stress induces plasticity or launches plasticity in cancer cells. It can be this plasticity captured by mathematical models, partial differential equations, that are not necessarily structured in space, but structured in variables that represent continuous phenotypic variables. And in particular, these models are compatible with optimal control methods and the design of therapeutic strategies. If you think of a drug resistance, the expression of drug resistance, as such a continuous phenotypic variable determining the plasticity of cancer cells. So drug resistance, or generally resistance in cell population exposed to an aggression, to an insult coming from the environment, has to my knowledge, at least, first been documented, but in the bacterial world, by Luria and Delbrück's experiment of 1943. So it consists of exposing bacterial population bacterial population in various petri dishes as exposed to phages. Phages are viruses that are able to destroy bacteria. And the question was, is resistance induced by the introduction of the phage environment, scenario A in this figure, or was it present, pre-existent in some clones, stochastically determined and selected by the phages, by the introduction of the phages, which is scenario B. And the experiments performed on a set of petri dishes representing a sort of probabilistic space, in fact gave the answer that it was always scenario B, that is pre-existent mutations before selection induced later after the pre-existent presence of these subpopulations of resistant bacteria, and not the complete induction by the phages in the populations. But I should stress that bacteria are not cancer cells in an animal, in a multicellular organisms. That is, animal cells in multicellular organisms, cancer cells, are much smarter than bacteria. And then came this very interesting experiment, also in petri dishes, but on populations of very aggressive cancer cells in a non-small cell lung cancer, in fact, of the species PC9, exposed to high doses of drugs. And this was at first the first panel that you see, then exposed to this high dose of drugs, 99.7% of these cells die. Only very few of them survive in this hostile drug environment that will be maintained in the petri dish for the whole course of the experiment. These are what people call drug-tolerant persisters, or DTPs. And in the same hostile environment, if you wait for some time, in fact, a long time, then 20% of these DTPs resume proliferation, becoming drug-tolerant expanded persisters, DTEPs. Total reversibility to drug sensitivity may be obtained by drug withdrawal. You must consider that the drug bath is maintained for the whole duration of the experiment, but if after some time you withdraw, you rinse the drug from the environment, from the petri dish, then you regain little by little, this takes time, in fact, the initial situation, and you can go on and on again with the same results, which means that resistance to the drug is something that is not genetically established, or at least it's not permanent, it's variable. And what induces us to think that it's not genetic but epigenetic is that inhibition of this enzyme, epigenetic enzyme KDM5A, which is a demethylase on the K, which is a lysine, it blocks the emergence of DTPs. So there is induced by these experiments the idea that resistance can be labile, can be reversible, which was a discovery of 10 years ago, but which was new for many, many people. And now it seems that among biologists, these drug-tolerant persisters are a very fashionable topic, and we hope that next year there will be an international conference on these drug-tolerant persisters at Embo, Heidelberg. If some of you are interested in it, I may give you information about it. Well, now,
Jean,
yes.
sorry because I see that you are going to start the more mathematical part, but I
Yes.
think that this is something very important which you presented now, and maybe we should have not maybe discussion, but just to summarize what you say to make sure that we understand correctly. So traditional point of view is that cancer emergence and cancer, and then the emergence of the resistance to drug are usually associated with genetic mutations. So what you say, yes. So what you say now, that this is not necessarily the case.
Not always the case. Yes.
Not always the case. So what between always, not always, and all this, all this scale of frequency of this event, where we can put all that. So is it just an exceptional situation where this epigenetic changes determine drug resistance, or it's more frequently or always? So you see what is my question is?
Yes, I see.
So how often this happens? So between genetic and epigenetic reversible mutations.
Well, to be sure that it's an exceptional or current phenomenon, we should have to consider many phenomena. What I think personally is that it may be a very current situation, but that it may also induce a situation in which genetic mutations are favored. And this situation may be transient, it may be reversible, it may be irreversible. This is difficult to know. What I showed you is a situation in which it is transient and totally reversible. But there may be other situations. And in fact, there are changes in, how should I say, the point of view between epigenetic and genetic changes in the minds of biologists. And it seems that some epigenetic changes can induce genetic changes. And this has been documented, but not so much. I think that it's a big challenge for epigeneticists to document this. So where the index should be put is difficult to say, in fact.
Yeah, so but what I just realized that I am not sure that everybody in our audience knows well the notion of, well, I'm pretty sure that everybody knows about genetic changes, but I'm not sure that everybody is so familiar with epigenetic. So if you can shortly explain what this means, please.
Well, what is genetic? What is a genetic mutations? Is a change in the code, in the genetic code, but to be expressed, genes are dependent on these genes to be either silent or expressed. And when you graft terminals such as methyl or acetyl, when you demethylate or methylate, acetylate or deacetylate these genes, then you can silence or overexpress them. And this is exactly what epigenetics is about. So a gene may be present, and I think that all the genes that give rise to resistance in multicellular organisms, in our organisms, are still present, but they are usually silenced. And in cancer cells, such genes, such methylations, for instance, can be suppressed. That is, demethylation of genes can make these such resistance genes to be expressed. And such epigenetic enzymes, such as KDM5A that I mentioned here, can do this job. And what makes plasticity present in cancer cells, but also in some situations in normal cells, but rather exceptional, in development or in wound healing, for instance, but not usually in physiology. It's a matter of pathology, usually. And this is how genes that are normally silenced in our genome may be expressed in cancer cells. I hope I have been, I have clarified some things, but people who would like to know more should interrogate, I don't know, Google or the internet about epigenetic enzymes.
Yeah, sure.
How they can
Yeah, I think that it's now more clear, but just to summarize, epigenetic means that something which make genes be silent or expressed. So
Yes.
these are some molecules in the cell with the same DNA. There are no genetic changes, there are no mutations,
Yes.
but there are some molecules in the cell which can make some genes to be expressed, to function, to begin to produce proteins, or to stop them. This is what means epigenetic, and what Jean explained us, that these epigenetic changes can be very important for cancer development and for the development of the resistance to drug. So I think that, yes. Okay, sorry for this interruption, please continue.
To mention another point, it is that genotype is not phenotype. So phenotype is what is the expression of the genes when they are expressed or silenced. This is what makes normal cells to be completely different, because development and differentiation physiologically is all a question of epigenetics. That is, how phenotypes, how cell types will be expressed differently and make, for instance, skin cells or intestine cells or neurons, etc., from exactly the same genome is a matter of epigenetics, that is, of differentiation from the same genome. And so I think that is important because it relates cancer to development in a very concise way. So let me go on. I will try to speak a bit of mathematics, then I'll go back to evolutionary biology, then only to mathematics. So when we saw with the two postdocs who are now associate professors, Tommaso Lorenzi in Turin and Rebecca Chisholm somewhere in Australia, I proposed them to design a model of a cancer cell population with two structural variables representing viability and fecundity. And I took these words, in fact, from ecology. And I think that somebody who is in St. Petersburg now, Denis, I don't remember his name, but I think that people among you may have attended his PhD defense, showed me that this is a very common way of speaking among ecological populations. So viability means, for me, expression level of a survival potential phenotype, and fecundity, expression level of the proliferation potential phenotypes. So it's a two-dimensional phenotype, X, Y, and from this we could, or at least mainly Rebecca Chisholm and Tommaso Lorenzi, could design an interesting reaction diffusion advection model, which was able to present all the characteristics of what was observed in petri dishes in this article that I showed you. So there is an advection term in this equation here in red. There is a reaction or selection term here in magenta, and a non-genetic phenotype instability, which is a diffusion term, which can be represented also by a mutation or epimutation integral. So with this ad hoc model, or with an agent-based model, this was Rebecca's work, we were together able to answer the three questions that I put into our model, that I put as a mission to our model. Is non-genetic instability, that is a second order term, crucial for the emergence of DT EPs, that is stabilization? What can we expect if the drug dose is low? Because what everybody should know is that when you induce drug resistance, usually in labs, then you use little by little growing doses, and finally you stabilize the phenotype, and you may have attained the genotype level by, or at least you have very well stabilized the phenotype as a resistant phenotype. So what can we expect if the drug dose is low? The situation in labs that are accustomed to usually to study resistance. And could genetic mutations with a kernel integral with a small support, that is a small mutation integral, generate the same situation? And we considered two scenarios, and the answers were very clear with our models. The Laplacian term is absolutely, whatever the scenario, necessary for the emergence of, the DTPs. Low drug doses result in DTPs if DTPs are present, but not, they cannot induce DTPs. And genetic mutations can never induce the appearance, the emergence of DTPs or DTEPs. So this was, in a summary, what we published in Cancer Research in 2015. Also, now it's a question of vocabulary, because among biologists, it's very important to distinguish between drug persistence, drug tolerance, and drug resistance. As I write here, I will use in the sequel resistance as a generic term for all these terms, but drug persistence for biologists, it means that a prolonged drug insensitive subpopulations, not necessarily the whole population, can be identified. Drug tolerance if the whole population is considered by transient treatment escape, and resistance if it is genetically established. So these are biology terms that we must have in mind. But for us mathematicians, I will use tolerance as a generic term, all the more so as we have hints that stable persistence may by itself promote, either by a selection on genetically unstable cells or by another mechanisms, irreversible drug resistance by mutations. And as I said, there's a small hint that silencing by methylation, that is an epigenetic intervention, makes single nucleotide C2T mutations on the DNMT3A locus highly probable, and training more and more epigenetic alterations, and finally genetic mutations. So this is difficult to obtain, but this article, in fact, is entitled, if I remember well, Genetics or Epigenetics, Two Sides of the Same Coin. So drug tolerance, to make this a long slide short, will mean tolerance or resistance the same for me. But maybe I will not go in the detail of this, but something important is cellular stress in cells. And cellular stress, in fact, is a cell state in which a cell endowed with some plasticity can launch a variety of responses to environmental insults, such as a drug, hypoxia, reactive oxygen species, radioisotope, etc. And a priori, we must suppose that these responses are should be different. So this induces us to think that, and this was more or less documented in this paper by Amy Wu in PNAS 6 years ago, cancer cells should overexpress cold genes, very ancient genes that are never substituted, and that could be make plastic cells able to launch different answers, responses to cellular stress of different forms. And to me, these speculations refers to the atavistic theory of cancer that I mentioned, and I will try to develop now. In summary, it states that cancer is a very primitive state of multicellularity in which multicellular organisms are not yet cohesive, but all cells are very plastic, and they are trying to put at work the basis of multicellularity, which is mainly cooperativity between cells. And in fact, in tumors, you can see tries about establishing some cooperativity. But of course, it never works because what has been done in the course of billion years cannot be achieved in the course of even a human disease. But the idea is that cancer cell populations divert these genes, this instability in gene determination for their own benefit. So this is what is presented usually in the Atavistic.
Sorry, Jean, I have another question about this atavistic theory of cancer.
Yes.
Because this is also important, and maybe we can spend a couple of minutes discussing it if you don't expect to do it later. But as far as I understand the theory, this means that these cancer genes are already present in the organism, and
yes,
and in the
cancer
stress
cells are our cells. In fact, they are cells of our organisms. So all the
no,
genes.
I mean that this theory does not imply genetic mutations. In fact, all genes are already there,
absolutely.
but due to environmental or other stress or other situation, so the cell returns, begins to express some genes which are already there without mutations and which were silent before.
Absolutely.
But then where are mutations there? Because we still know that rather often cancer is and malignant cells are related. They are related to mutations. So how these two things are connected to each other? I mean, this mutations and atavistic theory.
Mutations, in fact, should appear usually firstly on the genes that code for epigenetic enzymes. And I will try to put forth a few arguments about this. But then if these epigenetic enzymes and the genes that code for them are genetically mutated, then they will not be able to design their work of silencing these genes that are that should not be present because they involve plasticity, and plasticity is not compatible with the stability that is needed in our multicellular organisms. If we do not want to decompose at every moment, and which is in fact what is happening in cancer little by little, we have to have a stable multicellular construction in our organisms, and this is the work of epigenetic enzyme that methylate, principally, mainly, these genes that are normally responsible for plasticity. And these genes are not silent during development, during early development, and they are also not silent during some kind of wound healing, or even in some very clever animals, such as lizards or axolotl, that are able to rebuild completely either one tail or even one limb. So something has been regained of plasticity for such a construction, for such a design of a missing limb. But if the lizard or if the axolotl is still alive, then it has been immediately after that re-silenced by the epigenetic enzyme. So there is a short window in physiology during which these epigenetic enzymes that give us, again, plasticity are activated. So this is also a matter of physiology, but in very rare situation. Whereas in cancer, because these epigenetic enzyme genes are mutated, in fact, and this mutation that happens in cancer usually begins by these mutations of an epigenetic enzyme genes, then because of that, all the other mutations can begin. So have I been clear enough? I hope so, but I will try to document it a bit in the next slide. Well, this reverse evolutionary framework, which is present in the atavistic theory of cancer, I mentioned already that it has appeared in the development of multicellularity, which happens, in fact, during this period of time, during the evolution on the time scale of billions of years, during about 500 million years in the evolution of multicellularity, of cellularity and multicellularity, which corresponds to the oxygenation of oceans and atmosphere. And this has a biochemical explanation that I could answer this question if it's posed. But this has been documented in phylostratigraphic analysis and by Domazet, Locho, and Tautz, and investigated by Triggers more recently. So to make to give an answer to a question that has not been posed, the oxygenation of oceans and atmosphere is essential for the synthesis of collagen, and so multicellularity cannot exist without collagen or similar proteins. And so this is the usual explanation of this time scale of 500 million years during which multicellularity develops. And as I said, in evolution, it should come first by perturbation, alteration of epigenetic control of differentiation, then cell differentiation and division of work, and finally proliferation and apoptosis. Because what has been documented in Hirsch and in this paper in Nature Communications is that it was documented in fresh blood samples in leukemic patients. The first mutation that appear are exactly what I mentioned earlier. First, epigenetic control on differentiation, then cell differentiation and division of work, but finally proliferation and apoptosis that are the most soundly designed bricks in the multicellularity in the multicellular organisms. That is, you can see it as a rocket. Proliferation and apoptosis is the most sound, the first basis. Then you have to have a cell differentiation and division of work to have beginning of cooperativity and compatibility, and epigenetic control of differentiation and proliferation comes last because it is the finest control that makes the whole multicellular organism stable and coherent. And I again stress that cancer is a disease of multicellular organisms, not of single cells, never. So if we can have access to the phylogeny of the multicellularity genetic toolkit that has been developed in this period of 500 million years, then we could also have access to the genes that are altered in cancer and see how we could repair them. Again, to show you that there have been other explanations by metaphoric representations. This is a classic Waddington landscape, which I present to you because I think it's important to have in the mind that it consists of the differentiations that lead to a multicellular organized construction, but it starts from the zygote, that is from the same genome. All cells in a multicellular organisms have the same genome, but differentiation occurs in this metaphor given firstly by Waddington, and this is a matter of epigenetic enzymes. And this, in the reverse of this landscape, you have, in fact, the connection between the genetic the genetic and epigenetic enzymes that constitute these valleys and barriers, epigenetic barriers that give rise to this representation, which in fact is also a concern in systems biology. And some of you may have heard of Sui Huang, who says that nothing in evolution, not only what I think it was mentioned, nothing in evolution can be understood without the light. Nothing in biology can be understood, without the light of evolution. And Sui Huang said that nothing in evolution makes sense except in the light of system biology. So this is his point of view. And this has been also documented in an association between Sui Huang and Tariq Enver to try to show how this differentiation tree occurs in multicellular organisms. So at the top of this, you may have either the zygote or the first hematopoietic stem cells, and then it depends on the tissue, in fact. And then you have this tree of differentiations, which may be documented at bifurcations by differential equations, ordinary differential equations with stability or instability. And I propose another metaphor. These were metaphors, of course, which is related to more to at least how should I say, differential geometry, but not in details, which sees multicellular organisms as a fiber bundle. A fiber bundle constituted of a base, fibers, and what makes these fibers coherent between them is what I call the cohesion watch. So this should be developed with colleagues, but it's still in its infancy, but I think that the idea makes sense. That is at the basis, which is represented here, which may be of anatomic nature, you have the body plan, which is documented in papers by evolutionary biologists. Then from this body plan stem the fibers that are the cell differentiation trees. And of course, they must be connected between them if you want to have a coherent and stable multicellular organism. And at the end, you get at the rim of this construction the different cell types that constitute the multicellular organism. And all these three necessary things together may be considered as part of a Borromean knot, which was suggested to me by a colleague. So body plan, fibers, and what is less documented, which is a cohesion watch that makes, in fact, it's constituted of non-cellular connections between the fibers. And this, as far as I know, also a meaning in the differential geometry, which is connections between the fibers. Well, this is in its infancy, but I think that it's a promising way also to go beyond this metaphor. And now, if I can go on, yes, I think I can go now to modeling. But maybe, yes, I think that, no, sorry, I have missed something. I have no access. Yes, a weak one, but yes. Now I will dive more in mathematics, if you allow me to say so, and give time for discussion about evolutionary biology and mathematics at the end, if there's still is time. So what we have in mind, not in this last metaphor, but in all the other representations of cell populations, and in particular healthy and cancer cell populations that are exposed to drugs or other insults, is the fact that they are able to evolve by changing a hidden phenotype. And this phenotype or structure variable at the most elementary level is a trait, a one-dimensional trait that may change under the pressure of an environment. So the variable is a population density, depending both on time and this structure variable. And if you sum over the total of this, which may be at the elementary level of 1D dimension, which may be just an interval, if you sum over this population over X, then you have the total population, which I call rho of T. And if you consider at each time T, the quotient N of Tx divided by rho of T, you have a distribution of the trait in the population, and you can investigate its asymptotics. So these are the two questions that we can pose. Can we investigate the evolution in numbers of the individuals constituting the population? And can we investigate the asymptotics of this distribution in the population and see it evolve, in fact? Note that space is not necessarily a relevant structure variable when studying drug control, but it may also be associated. This makes more complex models, of course, and we have designed some of, but with just two variables, one structure variable representing phenotype and one-dimensional space variable. And this has been published in a paper in 2015, but I will not mention this. So these are the two questions. And if we use a non-local Lotka-Volterra integral differential model, which we will do, which I will do in the sequel, then the equation is very simple. It is this one. And you have here the instantaneous R of X rate of proliferation and the instantaneous rate, in fact, instantaneous, but it does not depend on time. The instantaneous rate of death is here d of x, and the death rate, the total death rate, depends on time because it multiplies the total population. So this is a simple Lotka-Volterra model with a non-local distribution of the death term, if you want. And so we can study such Lotka-Volterra non-local time and study convergence in time. And if you give a bounded variation BV hypothesis, then it's relatively easy to show that such a model converges to in time to a value, to a plateau, which is the maximum of R over D on 0, 1. Remark that this depends only on X. And that is the smallest value rho, such as this quantity is negative. So this is a simple simulation illustrated the convergence in time. But now you can also consider what I call not convergence, but concentration in phenotype. And here you can see the evolution of a population of cells with respect to this phenotype, the distribution of this phenotype, finally concentrating on the phenotype X equals 0 in this simulation. And so the complete theorem is that rho converges to a plateau. The distribution of n of t and x concentrates on this set, and we can never obtain theoretically that it's always a singleton, but in simulations, it's, and this is still under exploration and investigation, it's difficult to obtain anything else than a singleton. So if you add at least this hypothesis that this set of concentration is reduced to a singleton, then the distribution of the population converges in a measure set on this measure, which is a Dirac with a content, which is this plateau. And
sorry, what is singleton?
A singleton is just this set, supposing that it has just one element. A singleton is a set constituted by one element.
Okay.
In set here.
Yeah, I think that mathematically speaking, it may be related to Dirac delta function.
Yes, yes. But if you cannot obtain this, then you have just the same, the set that, I don't say that it's easy to prove, but it's if it's not a Dirac, it may be a Dirac-Cum, or at least a sum of Diracs that constitutes this set. But sorry, what did I do? Presentation, maybe. Maybe I should again share. Not, I don't want to.
Oh, it seems that you have slided.
Yes, I know.
Yes, so try to slide it
yes.
left.
But this is a thing that I don't know how to do. If I do this, it's not enough. So I'm very sorry for this. But yes, I know what I have to do. It's here and then aperçu here. Yes, sorry for this interruption. So this is the first things, elementary things that we can prove. And what Camille Pouchol in his PhD thesis has proved is that, in fact, it's not necessary to do this because BV hypothesis is enough, but it's very interesting to show that if you use a Lyapunov functional, then, which is this one, in fact, where the weight is nothing else but the inverse of the death rate, then this Lyapunov functional shows both at the same time convergence and concentration, because the derivative of this Lyapunov functional is, in fact, it can be proven, divided into two quantities. This one, which converges to rho infinity, that is convergence in time, and this one that converges to 0, which proves that at least the support of this measure set, is of measure 0, which does not prove that it is a Dirac-Cum or a singleton, but at least it proves that it's of measure 0. So, and this argument will be used in the sequel when we will consider two-dimensional setting. And this comes here at this two-dimensional setting, which was in fact considered already in 2015, 13 and 15 in this, what I call pedestrian optimizations of the study of this setting, in which you consider two populations of cells, a healthy cell population and the cancer cell population. They both follow the same non-local Lotka-Volterra model, but you have also points of control for two drugs. Here, the cytotoxic drug addresses an artificial death term, which is added to the non-local term, but here it's local. No, it's non-local. Sorry, but it's an addition to the proper Lotka-Volterra non-local term, and you have also a point of control on the proliferation, not on death term, but on the proliferation term, by dividing the proliferation rate here. And these two terms, U1 and U2, represent drugs. They can be studied at first with constant concentrations, but later in an optimal control setting, they will be variable. So what is IH and what is IC? In fact, they represent the environment induced by the mix of these two populations with different coefficients here, ACH, ACC, AAH, AHC, which represent the impact of the two populations, rho H and rho C, on the environment. And these are related to in fact, how should I say, the niches of these two populations, in as much as they can considered as having different nutrients. Well, this is a point that can be discussed biologically. So in this first paper, with Alex Lortz as a first author, we proved by just proof of concept that it could be optimized at constant doses. So, okay, but then we went further. There are some noises.
Yeah, so sorry, there is music there. Johan, is not it in your, because Johan, you have your, no, you switched off your microphone. Okay, so let's try to continue.
No problem. So if you use asymptotic behavior with constant controls, U1 and U2 are constant here, then you can show by generalizing this Lyapunov functional to two populations, and this was one of the main results of Camille Pouchol's PhD thesis, then the same result holds. That is, you have convergence and concentration is in fact on the support of the probability measures that are here, that I introduced earlier. And as regards convergence, then you have also convergence to two plateaus, one for the healthy population, healthy cell population, and one for the cancer cell populations. So you have the generalization of what was a simple setting for one cell population to these two populations, which enable us, in fact, to consider the impact of the two drugs on the two populations. Because when you consider therapeutics, you give anti-cancer drugs that always address not only cancer cell populations, but also healthy cell populations with different modes of impact, of course. But you can never expect that an anti-cancer drug will affect only the cancer cell population. It is hoped that it will affect mainly the cancer cell population, but not, you can even exclude that it will affect also the healthy cell population. So we have now a setting in which we can try to optimize the control by U1 and U2 being variable. And the basis, as I said, is the Lyapunov functional. And it's the same as the one I presented. in fact introduced by Pierre-Emmanuel Jabin and Gaël Raoul in 2011, I think, with this weight, which is exactly the inverse of the death rate. And so this works. And the cell killing strategy is here shown, and with that is, of course, these coefficients are artificial. They are not identified so far, but they are supposed to preserve healthy cells. That is, we have introduced coefficients such that the cytotoxic drugs is mainly active on the cancer cell population, and to a much lesser extent to the healthy cell population. So the optimal control problem is finding the control, depending on time, in some L2 space, minimizing in fixed horizon T. That may be studied varying, but it's in the optimization problem, in the optimal control problem, it's fixed. And the objective function is just the integral of the cancer cell population that must be minimized at the end of this fixed horizon 0T. But we have also constraints linked to the state, which are trying to represent the fact that we must preserve healthy cells. And one constraint is that the healthy cell population must not go below a given percentage of the initial population, and also that the total healthy cell population compared to the ratio to the total population, healthy and cancer, must be kept above a given level. So this is optimal control with constraints. And before I give the main results, I must stress that it's also setting that gives you the idea of what should not be done, absolutely not be done. That is, if you want pessimal control, the opposite of optimal control. That is, if you give high doses of both drugs, in a given time, you always observe that cancer cells concentrate on a very aggressive phenotype. I have not said this, but between 0 and 1, X represents the aggressivity, that is the resistance expression of the cell population. And the closer it is to 1, and the more resistant is the population. So what you can see also is that at the beginning, the cancer cell population decays, but after some time, it regrows and much above the initial level. And it is catastrophic for the healthy cell population, and also for this this subpopulation that we may consider, which may be considered as the sensitive cancer cell population. So catastrophic for the effect of these two drugs. This is a pessimal control. Now, if you see what the optimal control theorem gives, the proof being, of course, using the Pointfier gain maximum principle, but I will not show you. Maybe either Emmanuel Trella or Camille Pouchol himself could present this in a forthcoming seminar. You will have maybe this opportunity to listen to them. But the result is the fact that the optimal therapeutic trajectory in U1, U2 in L2 spaces consists of two parts. A long time part with constant control, but you will see in simulations, consists of doing nothing or almost nothing for cytostatic drugs, at least cytotoxic drugs. And the short time part, which is a boundary arc, a free arc, and another boundary arc. So this is maybe what is classical in optimal control on ordinary differential settings developed, for instance, by our colleagues Ursula Lentzewicz and Heinz Schettler, but they use mainly, as far as I know, only ordinary differential settings, and here it's partial differential settings. So there is a singular arc and bang-bang control for the rest of the time. And so these are the simulations. If you see the solutions are here for the cytotoxic drugs, and there U1, and there U2 for the cytostatic drug. So the idea is, firstly, do nothing with respect to cell killings drugs, which may be counterintuitive, and in fact, it should be considered by therapeutics, by oncologists, only, I think, if you have first given a drug administration that stabilizes in a first place the cancer cell population, then comes naturally the question, what should we do now? What should be done next? And the answer is never go on with giving the same high dose of drugs, but wait for some time, then leave the population being able to be sensitive again to the cytotoxic drug, because the long administration, which has decreased this cell population prior to this optimal control problem, has also, it's unavoidable, it's inevitable, rendered this cancer cell population little by little not sensitive but resistant to the cell. So we must give it some sensitivity again. And so this is what in the clinic they call the drug holiday. Drug holiday, which is rather current now in the clinic. And after some drug holiday, they give the same drug again. And what we advise here is to give it for a short time at maximum dose, and then stabilize it at an intermediate dose, which is difficult to determine, but not at this high dose. And for the cytostatic drug, it's also or in fact, it's completely or almost completely bang-bang. First, you give a dose of a cytostatic drug which just controls the proliferation. Then, at the time when you give the highest dose possible, then you can maintain for the cytostatic drug this high dose, because it will not induce resistance. The hypothesis here is that drug resistance is due only to the sensitivity to or insensitivity to the cytotoxic drug. But of course, you could change the coefficients and obtain the fact that you could also have resistance to the cytostatic drug. And this would give different results. And so what is shown here is if you vary the horizon, well, you obtain the same solutions with the results that are just the same, convergence of these cancer cell populations to almost zero, maybe zero if you can go below one cell, and you can also have preservation of the healthy cell population above a given prescribed threshold. So this is what we advertise for optimal control on cancer cell populations by using two different drugs, which one is among which one is a cytotoxic drug that is, in modern words, a chemotherapy, and the other a cytostatic drug, which may be, for instance, what is the name? Tyrosine kinase inhibitor, for instance, just a drug that affects proliferation, but that at low doses or medium doses at least will not kill cells, which is different from cytotoxic drugs. So this could be used in the clinic, but so far we have failed to find teams of oncologists who were sensitive to our proposal.
But Jean, at the same time, you mentioned that this drug vacation is used in clinics.
Yes, yes, but with I think it's with the idea that it's only to give the other cells, not the cancer cells or the healthy cells that are concerned by or at least only the healthy cells, the possibility to grow on again. Because you may know, for instance, when you go give a chemotherapy, what is also a limitation is the fact that it endangers the populations of hematopoietic cells and stem cells. So if the general conceptions among oncologists is that you have to give a window to help the healthy cell the possibility to grow on again, because otherwise you would kill the patients by completely killing the hematopoietic stem cells. This certainly is true, but we contend that also it has the effect, this drug holiday, to let the cancer cell population be sensitive again, which is generally ignored in oncologist teams. Well, the fact that also we have to confront the clinic should make us sensitive to the fact that they usually give periodic or almost periodic strategies. And for this, we have done some comparisons, and it seems that the second strategy it could be, that is better than the periodic strategy and adapted close to periodic strategy by controlling the singular arc rho H before going back to the drug holiday strategy. But this has not been explored much more than these few simulations, because it would make sense only if we had work contact with a team of oncologists or team of animal experimentalists, which so far we failed to obtain. So this was the point to which we were. And now I will just mention in the last slide work underway, last slides, which is important, because I think that bet hedging in cancer cells using this cellular stress that may launch a different strategy, which is a kind of thing that has been considered among different theorists, that may be completely different, but exactly as people in the stock exchange market do bet hedging, in the same way, when there is some cellular stress, we think that there could be different strategies launched at the same time, and they should address different phenotypes to be preserved for the population. And here we are exploring with a PhD student, Frank Alvarez Borges, with Jose Antonio Carrillo and others, but mainly the three of us, such a reaction diffusion advection setting with a new idea, which I took, in fact, it's not very original. I took from the Kane-Toad equation that was considered by Vincent Calvez and Émeric Bouin, which consists of affecting here second-order derivative with a term that depends on a structure variable representing plasticity. That is, you have two competing here phenotypes, which may be fecundity viability or fecundity motility or German soma. And you have another, a third structure variable, which represents plasticity and which should impact also the second order derivative here. So this is presently documented by this article submitted. You can have access to its version by clicking on this. And so it's another way of seeing things before control. And in fact, here we do not include so far control by drug D here. Here is cytotoxic D. Here is cytostatic drug D, another drug, sorry. But in fact, so far, we are just seeing, we have just proven, or Frank and Jose Antonio have proven that the model is consistent and converges before we can be able to study control, which will be for next step, I hope. Also about therapeutics and about this, you may have heard about immune checkpoint inhibitor immunotherapies, and I have just begun with another PhD student to design a model that I will not present you here, but it's a thing that must be considered, and there's plenty of space for modeling for this, because it's just modeling for immune checkpoint inhibitors or immunotherapies in general are still in their is still in its infancy from point of view of modeling. And also, this is even more in its infancy. My idea, if I go back to this metaphor of the wicker basket, should be, could we reformat the cohesion watch? That is, the thing that is disrupted in cancer is by mutations of these epigenetic enzymes. Could this be, which are responsible for the what I call the cohesion watch? That is what makes compatible and cohesive, cooperative the fibers, the cell fibers that go to the different cell phenotypes. Could this cohesion watch be repaired by attacking mutations, but only on the epigenetic side? If we could repair these epigenetic enzymes that I suppose are responsible for this metaphoric cohesion watch, then we could obtain non-killing cell strategies, which exist, in fact, in some very special cases. You may have heard of the CML, the chronic myelogenous leukemia, and the imatinib, and also a kind of acute myelogenous leukemia, which is APL, acute promyelocytic leukemia, which may be completely cured by inhibiting a particular molecule by a drug called ATRA, or transretinoic acid. But these are very isolated examples of what can be done to modify or to inhibit perturbations of what I call this cohesion watch. So this is maybe for a remote future, but having in mind that we could try to use such representation to design in the future non-cell killing anti-cancer therapies, would be a very big step forward. It's not unreachable. So by way of conclusion, I just state that new therapeutic tracks for fighting cancer disease, we should optimize their use, which is seldom done so far, by using optimal control strategies. I think that there is plenty of space to do this in many opportunities. As regards immunotherapies, which are so far just cell-killing strategies, because they are designed to induce immune cell populations to be boosted and kill cancer cells. So they're just other cell-killing therapies, smarter than just chemotherapeutics, but just the same. It's a thing that addresses the immune cell version, the immune cell response, which is not the whole of the immune system. I contend that the immune system is not only the immune response, it's something that should comprise what I call the cohesion watch, and which is responsible from the unity of the organism by making cell phenotypes, all cell types, being compatible and cooperative together. So this is a version of the understanding of the immune system, which is also in the ideas of cancer philosopher Thomas Pradeux, who was published and is still publishing things, and he's not limiting his views to the immune response. So the immune system is what makes the unity of the organism. And if we can repair such organism by acting on the things that make these things that are still to be discovered, but they are not cellular, they are certainly of the nature of the connections in this metaphor that he used, using a metaphor coming from differential geometry, then we could make a big step forward in therapeutics. So, as I said, not mere speculations. If we have these ideas in mind, we can be sensitive to observations that can induce us to consider such possible future strategies. These are references, and also I can give Vitaly and Anastasia, of course, my presentation. And in a short conference paper of last year, I have proposed this view of plasticity in cancer cell population using biology, mathematics, and philosophy of cancer. It's only a 6-page paper, so it's very general. And yes, я благодарю вас за ваше внимание. Простите меня за мой не довольно богатый русский язык. Можно быть, что в будущем я буду, я смогу представить вам разговор по русскому языку.
Большое спасибо, Жан, за это очень интересный доклад. Thank you very much for this very interesting, very profound presentation. So maybe we have some more time for questions. Please, if there are questions in the audience. And waiting for the questions from the audience, I can maybe start this discussion. Jean, sorry, we see only the top of your head, so maybe you can put your camera.
Okay, thank you.
Yeah, okay, it's better like that. Yes. Okay, of course, there are many questions to discuss, and of course, we will not time to discuss everything today, but just maybe 1 or 2 questions. First of all, I'm not sure to understand precisely the following point. So when you suggest your optimization strategy and this drug holidays, so because they will restore drug sensitivity of cancer cells. Do you mean here that all this, that it is based on epigenetic and reversibility of?
Yes.
This is based on that, right? Otherwise,
yes.
Why it would restore the sensibility, right?
It's of course assumes that the system is still reversible with respect to the phenotype. If it has led to mutations, this cannot happen. But as regards this point, in fact, I think that a way to do accord, but this will not make the optimal control strategy available more. A way to represent the possibility of irreversibility would be to put this reversible scenery within a PDMP, that is a piecewise deterministic Markov process, and Markov changes would be mutations. Another way of seeing this, which would be also more compatible with ecology considerations, would be to add space, because we know that, for instance, the Galapagos pinches of Darwin have differentiated, and now they are speciated, they are different species because of space separation. So if we also have a space, and not, well, it's easier said than done, then we could have a setting in which we would be more compatible with evolution. But of course, what I present here as an optimal control strategy is within this time during which you have reversibility.
Yes, but there is also another possibility if you have without reversibility, but if you have two competing clones of cancer cells, and resistant, well, one resistant and one sensitive, and resistant is less fit in the sense that if both of them compete, then it's sensitive which wins, right? And then you apply drug for it and you kill sensitive. So you see, it's another, it's related, but it's a little bit different.
Well, what people have usually done when modeling resistance and sensitivity is exactly what you say. It's a binary situation with a resistant and a sensitive two populations. But usually in biology, it's not this situation, except if you allow for mutations or space separation. So my idea is that to obtain this situation of two separated clones, then you must have some space, something that involves the populations in different settings. And you could think of a sort of Turing instability giving rise to two different spatially separated populations, but that could, with changing environment, allow one clone, that is one separated cell population from the other, being able to impact on the other. And this would amount to the situation you described. But I think that this should imply some separation to have this binary situation. And this could be done by introducing space.
Now, this is a very interesting question, and we discussed it a lot in the more ecological context, because this is what is called sympatric speciation in ecology. Sympatric, parapatric, allopatric. Darwin's theory of natural selection implies sympatric speciation without genetic or geographical barriers. And still, this is what he suggested, Darwin theory,
Yes.
sympathetic speciation. Then it was discussed during centuries that it was not possible because there are no genetic or geographic barriers. But probably it is possible. It is possible. And for example, well, I don't want to refer to our works, but we use this exactly. We model sympathetic speciation, no geographic barriers, no genetic barriers, but different species can emerge. So it's an ecology, but it is the same in cancer cells. They do emerge. They can emerge even without space separation. But okay, we will not go because I have, or maybe other people have more questions to your presentation and not to some other topics. And another question I wanted to ask about this very interesting metaphor you used about cohesion. But what do you mean more exactly about this by this cohesion? Is it kind of, for example, metabolic networks or what?
Yes, I think so.
Right.
Then it's not cellular, it's not the immune response, but somehow there should be correspondences by metabolic networks between different fibers. That is, in fact, between different cell types. How should they be identified? I think that I was pointed to a researcher whose name is Erickson, I think, and he has studied, I think, yes, these networks between different cell subpopulations, if I'm not mistaken.
Yeah, this is very interesting. Yes, indeed. Okay, so maybe I should ask about other questions in the audience, please. If there are questions, you can ask or you can raise hand.
Or if there are none, of course, I'm accessible by mail. And, I would be prone to send you the presentation. So
yes, please. In fact, we put presentations on our website, and of course, we would like also to have a copy of your presentation. So if there are no, yes, please, Alexei, please go ahead with your question.
Well, yeah, I have a very small question. I know that other people will try to incorporate drug poisoning into their modeling, into their models. Well, you talked about windows of the holiday, yeah?
Yes, drug holiday.
Well, to incorporate poisoning of the body with drugs and to incorporate it exactly to the functional which they minimize. Yes. Did you try to do this? Maybe is there any possibility?
I'm not sure to have understood your question, but the idea, in fact, to use these windows, this drug holiday, would be by technologically, I mean, by having a technological implant that would give by pumps the delivery of drugs to, and this exists, but it could be also at the level of days or weeks. It could be by just giving pills, but I think that it might be more efficient if you could have a catheter delivering the two drugs and with an optimized program deciding to stop or deliver the two drugs.
So to tune it more efficiently,
yes.
Yeah.
Yes.
Okay. Thank you.
This is what's sometimes called personalized medicine, or maybe now there are some new terminology. I don't know, but yes, of course, it should be patient adapted to each particular patient.
Yes.
Yeah. Thank you.
Okay.
Okay, then thank you very much, Jan, for this very interesting presentation, and we expect your next presentation in our workshop, which we'll have in three weeks. So thanks again, thanks to everybody, and we will meet in the next seminar. But I don't remember in 1 week or in 2 weeks we have our next seminar, and then we'll have the workshop. So everybody is welcome. Jochen, I know that you were not available for the workshop as speaker, but
Not
you are very.
for the last 2 days.
No, I'm talking now to Jochen, but you are welcome as a listener if you have time.
Okay, and so I will send to you, Vitaly, and
yes,
to
Anastasia,
please, my presentation by mail. Okay.
Yes. Okay. If it is not, if the file is not too big, then you can do it by mail.
I think it might be less than 10 megs.
Well, we'll see. If I receive it, it's okay. If I don't receive it, maybe Anastasia will receive it. 10, it's a little bit the limit of what our university server accepts.
Okay. So
okay. Thanks to everybody. Yeah. Bye. Yeah.
Thank you. Thank you.
Welcome.
Bye bye.
Спасибо.
Bye.
Bye.